Welcome to On Air, a podcast from the airr community. The community organizes and coordinates researchers studying all aspects of B and T cell receptor repertoires. The podcast is supported by the Antibody Society. For more information, please go to antibodysociety.org. This podcast has a special focus on clinical use of the adaptive immune receptor repertoires. We will look at how repertoires are currently used in the clinic and also discuss different opportunities where repertoires can be a great addition, the reasons why we are just not quite there yet, and how to overcome the obstacles. We are happy that you joined us for this episode of On Air.
Welcome to the episode number 12 of On Air, the podcast of the airr community with a special focus on clinical use of the adaptive immune receptor repertoires. Today we will discuss T-cell receptor-mimetic antibodies and immunoinformatics in general with Charlotte Deane from the University of Oxford. Hello, Charlotte.
Hello.
Charlotte is a professor of structural bioinformatics at the Department of Statistics and heads the Oxford Protein Informatics Group in Oxford. This podcast is hosted by me, Ching.
And me, I am Ulrik Stervbo. Hello, everybody, and welcome. So, Charlotte, one question that we always ask our guests is, how do you get into immune receptors in the first place? What is it that's so fascinating with these things?
So I was already an academic working in the area of kind of protein structure, protein sequences, all of those kinds of things, mostly protein structure, really had an interest in that. And I had done my PhD with another guy who was there doing a PhD at the same time. It's called Zhaoqing. And he, at that point in time, was working for UCB. And we met at a conference. And he started asking me some questions about antibodies. Now, I knew what they were, and I wasn't totally ignorant, but it is quite literally, that is how I got into them, because he asked me some questions that I was like, I don't know. And so I went to look and read, because that's what you do when somebody asks you an interesting question. And from there, I started to build up like a lot of understanding of them, but also a lot of interest in the fact that they were a really interesting group of proteins to me. The kind of, immunology, I don't understand it, but then I also don't think anyone does fully. I understand bits. And then the structures of antibodies are fascinating, and, TCRs and nanobodies, the way that they combine to many different things, but also actually they're really similar to each other. And it makes them a fascinating case study for someone like me who really likes thinking about protein structure and binding and interactions, because you both kind of have this lots of data, sounds really simple, actually really complicated relationship. So it's kind of how it built. But I blame GA for all of it, just to be clear.
So your background obviously is not immunology. It's all structural biology. So what got you into structural biology then in the first place?
So I did, and my undergraduate degree was in chemistry. And I very quickly became aware that what I was actually good at was working on computers, not in a wet lab. Right? There are too many stories there, but basically, trust me, computer was a better place for me, and I enjoyed it a lot more. And really what happened was, in my undergrad, in your kind of fourth year, so I did my undergrad at Oxford, your fourth year is a year-long project. And I looked at the projects that were available, and there was one which was basically about proteins and protein structure, and I thought it sounded interesting. And so that's the one I did. And that's really where it all began. It was a topic that I did not know a great deal about before that, but the minute I started doing it, I really enjoyed it. I enjoyed kind of I really enjoy the mathematics and computation of it. That's really what my group majors on. That's where I work. But I also, I think proteins are beautiful. I think antibodies are amazing. They're beautiful things to look at, and then the kind of the development of algorithms where you have to think about the three-dimensionality of them, I found exciting. And then how that connects to how the entire biological system works. So it built from there. I then went on to do a PhD very specifically in the area of kind of computational methods for sort of protein structure work, and just kept going, really. Just kept going and going.
While I was looking into all of your publications, I came across these wonderful tools and databases that you've created. Could you tell us a little bit about is it the SADDAB, as well? And now I'm forgetting all the different acronyms, but yes.
So, really, all of the databases in my group, I think it's important to sort of set this out, arose from the same kind of frustration that I think everyone has. If you are somebody who works computationally on this, or even, any type of this, one of the biggest things is you're aware there's a large amount of data out there that could help you, but actually it's all in a bit of a mess because it comes from different groups. It's publicly available, but it comes from different groups. It's not consistent. You can't kind of club it all together. You can't work out what you can actually do with it. And the more we're moving into this kind of machine learning age, the more important having the data collated, consistent, and easily accessible is. So it feels like an age ago. We published it in 2014. The first of our databases was SABDAB. And the idea there was this very simple thing, which was if you went to the PDB and you typed in the word antibody, you didn't get all the antibody structures in the PDB. You got some random collection of some antibodies, some other stuff, and loads of them were missing. So we thought, well, we should be able to do that. So we created this fully automated pipeline for collecting those structures because we wanted to be able to use that data. It now has about, I think, 8,000 antibody structures in it. It's fully automated updating collection. It also contains nanobodies. So there's about just over 1,000 nanobodies in there, 1,200, something like that. And it doesn't just collect them, it curates and presents the data consistently. So if you want to find out which of the antibody chains are in contact with the antigen, if you want them numbered with a numbering scheme, you can have all of that from that database. And all our other databases kind of come from that same kind of frustration. So another database we have is OAS. That's the observed antibody space. That has, I think, about 2.5 billion antibody sequences in it. And once again, this is publicly available data, I think with sort of over 80 different repertoires that have been sequenced here. But what we have done is taken that data and sorted, cleaned, annotated, translated, and numbered it. So it's there for use. And then another one of our databases is TheraSabDab. And this was a similar kind of thing as SabDab. We wanted to have a database of the immunotherapeutic variable domains. Actually, once again, quite hard to do because most of the information on them comes out in PDFs, which means you can go read about it. But if you actually want the whole set and to know how they relate to everything else, it's very hard. So that's got just over, I think it's about 850 therapeutics in it now. And it's a collection of all of those, but we connect it back up to our other data sets. So we connect it to SabDab, so you can see if these things have a structural representative or you need to model it or how close a structure is available to it and to all the other information we have. We also have a database of TCRs. So like SabDab is for antibodies and nanobodies, we have STCRDab, which is for the TCRs. Exactly the same idea. And then the most kind of recent addition to our set was something we built starting in 2020. And it was really because my group wanted to be able to do something useful during the pandemic. I think everybody felt like that. If you work in this area, it was like, how can I be useful? And one of the things that we're good at is building databases. So we created something called CovAbDab. And I think, I couldn't tell you exactly, it's got well over 10,000 entries in it, but it's got all of the antibodies or nanobodies which have been proven to bind to different parts of SARS-CoV-2. So, and where they bind and all the sequences and all the available metadata to allow people to do that kind of examination as well. So yeah, and we've got more databases coming because, as I say, I think it's a really important part of what we need as a community is really clean, consistent data that we can build off to do everything else we do. And since we're building it for ourselves, we put it out there so everyone else can work with it too.
So these are curated databases. And is it difficult to, I guess, harmonize all the different studies or inputs?
Oh, yeah. Yeah. I mean, every time we build one of these, what we're trying to build is something that's completely automated in its updating. Yep. And SabDab is fully automatic. But by fully automatic, what that means is it has an internal emailing system that emails us when it gets confused about something it's tried to get down from the PDB. But also, and if anyone's listening to this who uses SabDab or looks at it, if you find a mistake, tell us, because it contains mistakes. All of these do. So we really want to hear when people are using them, and there are errors, and we're really happy to collect them. But we're trying to make these as much as possible automatic, because otherwise, this is an academic group building these. They will die if they're not relatively automatic, because I can't, the funding to have software engineers sitting there constantly to keep these perfect just doesn't exist in an academic sense. So it's important to me to make them as automatic as possible, but also to be responsive to try and keep them as good as we can.
I was wondering about model antigens. So is this a problem in these databases?
So these databases, all of them have biases within them. That's part of why to build the databases, is because if you build them and you have as much of the data as possible, you can then select out from it things like a non-redundant set to try and remove the biases. But they are massively biased. I mean, I think, the STCR DAB, the TCR one, is probably more biased than many of the others because also it has much less data. I think we've got, 600 odd structures in that one. Whereas, like I said, you've got about 8,000 in the antibody one. So you've clearly got more of that going on. And everywhere there's a bias because people are working on particular areas. But that's, if you collate the data, at least you can see the bias, is where I start from. Doesn't solve it completely, but you can see it. So then you can start to ask questions about what that means for what you can and can't do with the data as you go forwards.
I guess changing topics real quick. Speaking of T cell receptors, you recently have a Frontier Immunology publication about T cell receptor mimetic antibodies. It's a pretty, although I guess to most people it's probably new, but I guess historically they've been around for a while. Could you give us an overview of what they are and what makes them so important or exciting?
So, I mean, in very simple terms, it's, could I make an antibody that pretends to be a TCR? And obviously, in general, our antibodies in our body are doing the exact opposite of that, in the sense that they don't want to bind to the binding sites of TCRs, because that would be self-recognition, and this would be very bad. So what we want it to do is bind to the peptide MHC, or it depends what peptide HLA complex. Yep. So, and just to be clear here, when I think about these, we are thinking about things that actually bind to that kind of peptide MHC bit, not just somewhere randomly on the MHC, because you do have antibodies who do that, and they're related to things like organ rejection and stuff like that. So this is a different kind of thing. This is, I'm going to do an impersonation of how a TCR binds with an antibody. And the reason people are excited about them is because it's a way to look inside the cell. So what an MHC, or depends when you call it MHC or an HLA, but what they're doing is they're presenting peptides from things that are inside the cell on the outside of the cell. So that means I've got an extracellular signal of what's happening inside a cell. And if I can recognize that, that means I can actually use that for many, many diseases. The obvious disease that people are a bit excited for this is kind of bespoke antigens for cancer cells. So it's this idea of you can actually think about doing that using this kind of technology and this way of thinking about it.
I know, at least clinically, there's soluble TCRs out there sort of against these anti-tumor associated antigens. What do you think is, I guess, the differentiation of the TCR mimetics versus just a soluble TCR?
Part of this is the kind of, if you think about the genetic background of TCRs and antibodies, you've got a very similar molecule here. But what we also know is that TCRs in their kind of natural state, they are not very high affinity in their monovalent binding. So making them high affinity or high enough affinity as a soluble TCR is already a challenge, because that's not, their natural state is much lower affinity than that. Whereas antibodies, that's where they go. You do display libraries of antibodies naturally that high affinity. The other thing we know about TCRs is they are relatively non-specific. Most TCRs recognize a very large number of peptide MHC complexes. So when you want to turn them into a drug, you've got to overcome both of those challenges. Whereas antibodies are high affinity on average, you can make them high affinity, and they tend to be specific. But of course, now your challenge is that peptide MHC is not the natural kind of target, and in fact might be an area where antibodies don't normally head for. So you're going to have to think about how you push them into that space. So that's really, I think there are advantages of doing this problem in both ways. But the reason people are thinking about these TCR mimetics, I think, are really around that kind of affinity or specificity that antibodies can achieve.
How do you get antibodies to look for peptides in an MHC?
So the general way people think about doing that, and this is why it's actually quite challenging, and when people first started doing this, it was very challenging, is, of course, you do a kind of standard library screen. So you have the complex you want it to hit, you throw in a load of antibodies, but of course then they don't bind where you want them to bind. Okay? And so there is a level of kind of trying to work out your specificity here to ensure that they are binding and that the peptide is part of that binding. Because if it just binds to the MHC, it's useless. There's a sort of slightly more complicated bit there as well, which is you also don't want it, you have to think, do you just want it to bind to the peptide itself? Or is it the peptide MHC? Because that is its kind of context that it would bind in. But if it just bound to the peptide, that might not work either, because it might not be a large enough binding site for it to be specific, and it might have other factors. And these peptides are only 9-mers. So, if you don't hit all 9 residues, which you won't, you have all sorts of other issues. So the skill here is really in getting something that binds to the site that you are interested in with an antibody. And that, I think a lot of people now are thinking about changing the way they do their library design at the beginning to ensure that it contains things which are likely to go for that kind of target, and that kind of way of doing it.
Do I understand this then correctly, that the target or the binding site for an antibody would be part peptide, part MHC, or just the peptide?
Yeah, most people have gone for that part peptide, part MHC as the binding site. And it's an interesting question about exactly what you should go for. I mean, partly because, well, one of the reasons that's an interesting question is related to how you can turn these into therapeutics. So we all have very different kind of HLAs, MHCs. Yep. And so if I make it very specific for one of those, I may not have a very large target audience for my medicine. And also, if I go further down the route that it's specific for the peptide as well, and we also all have different mutations that may be associated, say, with cancer. So now it becomes a very personalized medicine alongside that. There are people trying to think of ways of getting around that problem. So that's, I think, people have sort of said the optimal way of doing this is to bind the peptide and the MHC, because you want the context and you want that peptide so you know you've got the right thing. But as I've said, that causes other issues. So one way of getting around that is having small molecules that target peptide and then antibodies that target the small molecule plus the peptide in that orientation. Because it mustn't just target the small molecule, or it will just eat up the small molecule. But that allows you to do this kind of, larger scale docking, or know that you need that peptide even if you don't have the same MHC. There's a few papers where people have been successfully doing this kind of thing. So I think it's kind of thinking about it that way, that kind of idea of it. There is a big challenge here about it's a magnificently possible personalized medicine. But of course, if it's too personalized, it becomes a very expensive medicine, which adds another dimension as to what you really want to do with it.
So adding these small molecules will remove the dependency on the MHC then?
Yeah, that's, I think, one of the big plans there is to see if that's the kind of thing that they can do, is remove that dependency. So you can have that kind of differentiation and you can control things more. But that's, I mean, I've never done any work on that. It's just that people have published on that. And I think it's a really interesting development in the field.
Going back to a point you were making about the docking of these TCRs and TCR mimetic antibodies to the peptide MHC. In your publication, you were talking about how the differences between how, right, a TCR versus a TCR mimetic engages. Could you speak a bit about that and sort of what was interesting that you observed?
Yeah, I think, well, the first thing I should say that it really is observations in the paper. So I'd love to say we knew an answer here, but we only have 10 independent structural examples of TCR mimetics. We have a much larger number of TCRs, but only 10 of those. And because of where this work is done, it wasn't somewhere we could go and get some sequence examples and model them. There is no sequence data. There are 10 independent examples out there with structural information. And I think the way we started thinking about this was, well, TCRs and antibodies are really like one another. So it's kind of going to be similar, isn't it? They've got, if you think about the genetic background, the way they're built, there's the two chains, the heavy chain and the light chain, and the beta chain and the alpha chain. That's what this is going to be like. And I think one of my postdocs, Matt Raybould, put this really well. It's like they're actually two different Lego kits. So they don't actually behave in exactly the same way. It's like you've got two different sorts of Lego. So of course they don't plug quite like you think they're going to. So the things that we saw was that you can see big differences between TCR mimetic. Well, you can see obvious differences between TCR mimetics and antibodies. For example, it's a much more concentrated set of kind of CDR lengths you see. But maybe that's just because you've got this nice kind of flat binding surface that you're trying to go for, and anything that bound to that kind of surface would be constrained to those loop lengths. But then if I do the comparison to the TCRs, it appears that, the antibodies can orientate in a very similar way over the binding interface to those which we see with TCRs, but they can also do it very differently. You can see both. But the loops that they use to do the binding are just completely different. And this is really where the Lego set comes in. It's not like, the heavy chain, I don't know, CDR3 and the beta CDR3, they're very important for this and they do this, and we see that in both. What you see is it's just different. The overall shape of the complex looks similar, but the actual individual interactions from the CDRs, either from the TCR or from the antibody, are very different. And I think that, one way to say that is, if you think about the genetic background, we would all, because we'd go, well, we know what genetic background is and how these things are built. The beta chain of TCRs should relate to the heavy chain of antibodies, because they're the ones which have the VDJ recombination. The light chain and the alpha chain don't, right? If we look at how these things, TCR mimetics, structurally engage, sometimes you would say that is what's occurring, and sometimes it's flipped. So the light chain is behaving like the TCR beta chain. Now, that to me is kind of an indication of what I mean by the Lego being different, as in, genetically, I would have said, well, absolutely, it has to be like that. But in reality, not at all. That relationship doesn't exist. And so I think it's like the global properties are similar, but the exact properties within are very, very different indeed. And I think that's kind of what made it interesting to examine these. And I think as we get more data, will become more interesting.
Yeah, I mean, 10 antibodies is a small data set, but it's also interesting. It's hard to ignore the convergence.
Yeah. And it was interesting to us because, there were only, I think 3 years ago, there were 4 of them. Mm-hmm. So the number is going up where, when there were 4, it was like, I don't really know what that means. Now there are actually 11 structures, but only 10 kind of independent ones. So you're starting to be able to see that the patterns and ideas that are coming.
And of the 10, you mentioned some of them also approach the peptide MHC differently, sort of in a diagonal manner. Is that the majority or a unique subset?
The problem is that I'm just going to say that some do this because I think it doesn't really matter if it's the majority out of the 10. So we see that some of them follow what is called the kind of diagonal binding mode, which is similar to TCRs. But as I said, even if they are doing that, they might have flipped the chains. So in my head, they're back to front. Okay. So it's diagonally approaching, so it looks like a TCR. But then where I would have put the heavy chain is where the light chain is.
That's what you mean. Okay.
Yeah. So that kind of, it's that idea of globally you look at it and go, oh yeah, and then you look at the detail and go, oh no, that kind of feeling.
Got it.
I was wondering one thing with so off targets, if you know anything about this, of these mimetic antibodies. So what could that be?
Well, in the off targets, I mean, most of this has been done in the oncology setting. That's where people have been exploring this. So actually it's effectively, it could be, if you like, the wild type is your off target. So it's the unmutated peptide in some way or something similar to that. And because it's 9-mer in the binding site, it's likely that there are others. Because if you think about this, if it's a 9-mer peptide that's doing the difference, because I know you're binding to the MHC as well, but that can present many, many different peptides. So if it's a 9-mer peptide in there, you've got a small number of those side chains are actually facing to the surface. Not all 9 side chains point in the same direction. Usually it's like 1 up, 1 down. So we might have 3 or 4 that we can play with here. So that's actually a very small number of different contact points. So you have to be very specific if you're only going to hit that. And also we know that these peptides are held in a relatively small number of conformations. It doesn't appear that, they're being held between these 2 alpha helices in the MHC. And so there isn't only one conformation, it's relatively small. So it would be surprising if there weren't other things that were very, very similar. So making specificity very difficult. But I think the important thing about off targets is that Not so much whether there are off targets that exist, because I suspect for most of these there really are and there always will be. And perhaps even not even how strong the binding is to those off targets. What matters is how prevalent those off targets are. Because if they're really, really common, even if you only bind them very weakly, it's going to do masses of damage because of the way the system works and what we're attaching here. So it appears to be that that's the important thing, is if you're developing these, yes, we have to think about off targets, but what we're thinking about are anything which is, we need to try and ensure it doesn't bind to anything that might be common in the system, because that is what will do the damage, rather than kind of, oh, it binds very tightly to another thing. Well, if that thing is very rare, yeah, it's bad, but it's nowhere near as bad as weak binding to something that's very common in the system.
Another paper I found when I was looking through your publications was your analysis of and development of the therapeutic structural antibody database, and just a broad comparison of the sequence overlaps of these therapeutic antibodies with naturally occurring antibodies. And I found it really interesting how the differences that you observed, which is like the fully humanized versus the chimeric versus the humanized. And so do you think that's part of sort of an anomaly of how these antibodies were made, or more of the screening process, or some other hypotheses? Like why do you see these differences?
Do you mean in terms of overlap with observed natural antibodies, or?
Correct.
Yeah. Well, I think it's most of that. Well, first part of that is I was really shocked when we did that first study. So we did the first study on that ages ago, and we had about 240, 242, I think, therapeutics was what we searched for. And observed antibody space, our sequence database, only had 500 million sequences in it at that point. And 54 of those 242 had a perfect CDRH3 match in the natural antibody database. I always remember this number because I made the postdoc in my group go back and check it like 4 times. He didn't like me very much by the end of this. But because if you just do a rough calculation in your head, there are, depends who estimates it, but 10 to the 10 to 10 to the 14 are good estimates of the natural antibody space. All right? I've got 500 million. That's a drop in the ocean. Speaking as a mathematician, if I took 242 random sequences and asked whether I'd get any matches in 500 million in that size space, the answer is 0. If I then put on top of that those 242 are meant to be highly engineered, the answer becomes really 0. But it was 0 before that. It's 0, just mathematically speaking, chances of there being a match are incredibly low. So I find it really interesting that those matches exist, because it suggests that the antibody repertoire is either not as large as we think it is, which might be one answer to this, or it is as large as we think it is, but most of it is not used most of the time, is another way of thinking about it. That our careful engineering is also driving us towards natural things, which I quite like. So, what are you going to make? I'm just going to make a human antibody. Yeah, one that's in that person over there. It's a very expensive drug. And so I think that's kind of where I started from in that. And then the overlaps that we observed, I think, are really related. It is also related to where these things come from. The majority of the data we have in observed antibody space is human, and then the next largest is mouse. So if something comes from a background that's very different from that, they will have a different impact, if you see what I mean. It will play out differently. So I guess that's where it comes from in my head. It's kind of, I wasn't that surprised by the difference between human, humanized. That's kind of what I was expecting. What I was surprised was that they overlapped at all. I was expecting the answer. Literally, this was one of those things where I said, we should do this, expecting the answer to be nothing happens. And instead, it gave an answer that I think tells us a lot about what antibody space is like. It doesn't answer the question, but gives us a lot to think about in terms of what that sequence space is like and how it actually operates.
Okay, so if I'm understanding correctly, you were right, like I'm looking at the figure that has the heavy chain sequence identity between the different antibody types and whether they were fully human and humanized. So the fully human has, 80 to 98% overlap sequence identity, versus humanized has a bit lower, around 75 to 90. So you're saying that to you is still surprising, or that they have or is that equivalent, or is that different enough?
I think it's surprising to me that the overlap is as high as it is at all. I mean, as I say, in the paper we go through the mathematical calculation of what would you expect, and the answer is none. I mean, very low numbers. But the kind of, once you're in the kind of 70%, I mean, every therapeutic antibody, I think we did this recently, I think now every therapeutic antibody we can match its chain, and it's about 80% sequence identity to something in OAS. Yeah. But I would say that's just because antibodies are that similar.
And that's just the CDR3, or is that the...
No, that's the whole chain.
Oh, okay.
But that's a kind of, to me, that's unsurprising, because antibodies are just that similar. And we're now, I mean, 2.5 billion is still a small sample. What's more important in those numbers are the specific parts which are involved in binding, because the overall framework is going to be similar. You know, we want that to stay the same. So it is. So it's probably similar to something that's in the database. We know enough now to know that that's what we're trying to do. But it's the similarity of the CDRs that I think is the interesting part there. How similar are they? And, you know, are we actually making novel residues in those to make novel binders? And, you know, obviously we are to a certain extent, but I think what's available in the natural repertoire is much more powerful than perhaps we thought it was for these problems.
Yeah, I guess the 80% homology of the full chain isn't too surprising to me, but the 54 exact CDR3 matches, that's pretty surprising to me in terms of, did you happen to do the analysis of within that 2 billion natural antibody repertoire, like what is that? What are the frequencies of those CDR3s?
So I can't tell you for the 2.4 billion, because we did this work when it was much smaller. Okay. We've done some work since then, but I can't remember the numbers. But there were several where we matched it from more than one repertoire. I can't remember the number, but there were several of them we found more than once. So it wasn't an odd chance. It was found more than once. I mean, my argument is finding any at all is a bit of a shocker, because most people engineer the CDR3. That's what they think they're doing. And, you know, but we found them, and it wasn't just one or two. We found a lot of them, and several of them we found multiple times. I can't remember how many, but several of them we found in lots of data sets. So they're clearly a normal H3, if you see what I mean.
Right.
Lots of people just have that H3 in their repertoire. It's floating around. It's there.
Yeah.
For whatever reason.
Okay. Yeah, I find that really interesting, because one of the themes that, you know, we like to keep going back to is the idea of how do we leverage these public data sets, repertoire data sets, to understand drivers of disease, right? The antibody response, and then deconvolute the antigen. And then, so then the question is, are there public, you know, BCRs and TCRs that are disease relevant? And so it suggests that, at least for your work, that, you know, even from the engineering perspective, it's converging on nature.
Yeah. I mean, that, and just thinking about that kind of public repertoire stuff, one of the other things we've looked at is doing large scale modeling. So structural modeling of a lot of the repertoire sets we have in OAS. Now, obviously we can't model every sequence. I haven't modeled 2.5 billion sequences, but we've modeled very large numbers of them to try and say, all right, even if I can't see sequence overlaps, because people have done those studies, do you have sequence overlaps between different humans? And the answer is almost none. And the answer to that question is, well, I expect that, because if I've only got a sample of 1 million from each human or less, and they've all got 10 to the 10, the overlap you expect is very low. So then we did but do we have similar structural shapes between different repertoires? And we found that the answer to that was very clearly yes. So even if and this is, to me, is a kind of, wasn't a completely expected result, but kind of follows through from the general way protein structure works, that actually the convergence might not be as easy as we've got the same sequence. The convergence is, if you have a very similar structure with similar residues pointing out, it can do the same thing. So I think there is convergence in those repertoires, but we can see it at a structural level, but we might not be able to see it at, you know, the sequence level, because we can't sequence deep enough. You know, if I could actually see the 10 to the 10 in each human, maybe I would start to be able to understand how this works completely.
Sorry, still have so many questions. Another follow-up to that. is that off of the predicted structure, or is that actual co-crystal structures of?
No, that all has to be predicted structures.
Yeah,
You've got nowhere near
that's
that.
what I thought.
The data.
Just making sure.
No.
But yes, that's very exciting.
Yeah, and I mean, and of course that comes with a caveat that of course the predicted structures could be wrong. So it could just be a freak of the methods. I don't think we tried to demonstrate that we don't think that's the case, but it's hard to be completely certain about that. But I don't think it's the case that's what's driving those numbers there.
So not only do we start with hearing a little bit about your background, we also like to ask our guests, so what do you think the future will bring in terms of T and B cell receptors, especially for clinical or purely diagnostic purposes? So what do you think the future will bring?
I always think this is the most dangerous question in the world to answer because you're bound to get it wrong if you go too far ahead. And if you don't go anywhere, then what was the point in trying? It's absolutely clear that the power of, TCR mimetics, so the kind of they're going to be very powerful in terms like diagnostics, even if they may not be medicines. And I think they'll be powerful medicines as well. But I think the trend within all of this is the data and the increasing volumes of data are allowing us to become much more predictive about how we do everything. I guess something that's really dear to my heart is to move the whole field when we're thinking in the clinical world from the phrase antibody discovery to antibody design. And I think we are moving that way. And I really want to see that. And alongside that, I think we're getting a deeper and deeper understanding of how these things work and what they actually do and why they're important. So to me, it's that kind of movement towards being able to design, not discover. Don't go fishing for these things, but actually design them from the ground up. And that's a real combination of experiment and computation. That's about being able to automate processes. It's about a very different way of thinking about how we do everything. But I just see it as this inevitable rush that's hitting towards us, and we should all be going, wow, that would be so cool, and be part of making that happen.
Could you also imagine engineering T cell receptors, not just antibodies?
Yeah. And I think would be, it's sort of obvious to me that that's part of what will happen. When we were talking at the beginning, we were talking about that, why don't you just do the soluble TCRs? And I don't actually think that that's necessarily a bad choice. It has different downsides from trying to do a TCR mimetic for these kinds of things. So I would really hope that will be part of it. And I guess I go back to the Lego analogy. First Lego that's easiest for me to think about because we have so much data is the antibody Lego. Best place for data currently from where I'm sitting. But we're getting data in the TCR Lego. New Lego. Go and build it. It might be more like Meccano. I don't know. But, that kind of thing as we build through. And, you can think about that for nanobodies as well and all of these different things. But to me, it's kind of learn the Lego sets, be able to design these things, and it will completely change the way we think about doing clinical development for these. It will be amazing, the speed and the kind of accuracy and specificity that we should be able to achieve by doing that.
Wonderful.
Answers were absolutely excellent. We will return in 1 month's time with more thoughts about clinical use of airr sequencing. This podcast is edited by Abdul Aziz of the comedy podcast About Law. Thank you for listening to On Air.
This brings us to the end of the 12th episode of On Air, the podcast of the airr community with a special focus on clinical use of the adaptive immune receptor repertoires. The podcast is supported by the Antibodies Society. Please go to the website antibodiesociety.org to get more information about our sponsors. If you have any questions or comments, drop us a line at onair@airr-community.org or tweet us using the hashtag #OnAir. That's with 2 R's. The contact information is also in the show notes below.